for the Beckman Laser Institute in the fall. Of course, it's all remote due to COVID. And our first speaker is Nick Hedde. Nick completed his PhD in physics with a biophysics emphasis in the Karlsruhe Institute of Technology in Germany. And his thesis was light microscopy beyond the diffraction barrier in live cell studies. He graduated with highest honors. He then came to the United States, and he worked in Enrico's laboratory for fluorescence dynamics, and researched plane illumination microscopy, or what some people call, I guess, light sheet. Since 2018, about 2 years ago, he's been working in Wing Zhao's lab, doing particle counting and sorting devices. So today, Nick's talk is titled Optical Detection of Rare Space-Time Events for Precision Medicine. So thank you, Nick.
Thank you, Thomas Milner, for having me today and for the introduction. And I also want to thank the organizers, Richard and Sandra, for organizing this virtual seminar here today. And of course, I want to thank all of you who joined in. Some of you maybe from different parts of the country and also different continents where the time might not be the most convenient. So thank you. Okay. Yeah, so my talk today is going to be about optical detection of rare events in space and time for applications in biomedicine or precision medicine. So I want to start by introducing two general problems in biological or biomedical research. And the first one is the so-called needle in a haystack problem. And here the issue is that often we are trying to find or identify rare particles or cells in a large volume that could be, for example, pathogens like bacteria or viral particles in blood, that could be circulating tumor cells, but that could also be specific B cells that happen to produce the right antibodies that we're interested in. So tackling this problem, if you are thinking about the literal needle in the haystack, how do we approach this? Well, typically we are trying to exploit a property that only our particle of interest has and that distinguishes that entity from the rest. In the case of the needle, if the needle is, for example, made of some magnetic material, you could use a magnet to fish it out. Or if it's shiny and reflective, you could try to scan with a laser and measure reflection to try to locate. In biology or biomedicine, we have a similar approach. We try to find a unique property of our particle of interest, which is often the molecular structure. So for protein, you could, for example, use antibodies that specifically bind to the protein of interest. Or if you're trying to detect RNA or DNA, that could be the complementary part that then can dock and identify that sequence. Contrary to the needle in the haystack, we cannot actually see our particles of interest by eye. So what we usually need to do is to attach some form of marker to highlight the particle. And for the purpose of this talk, we are going to be using fluorophores, fluorescent markers to label our structures of interest. So this problem is relatively straightforward. However, it can be, in reality, it can be very difficult to solve, as you'll see later. Another issue that's often in the field referred to as the school bus problem, is that some events can be rare in time and thus hard to identify. So we could imagine, let's say you are an alien from a different planet and you're given the task to find out what a school bus does on planet Earth, well, you would probably snap some pictures from orbit of the surface of the Earth, similar to what Google Maps does. And then most likely you will find that, well, what are school buses? They are these big yellow boxes, and they are all stacked next to each other in a parking lot. And they basically just aggregate there, they are static and they don't do anything. Of course, we know living in the society that this is not the case at all. I mean, they are very mobile and they are used to transport, to take kids to school and take them back home again. But this happens in a very narrow time frame, which we are most likely to miss if you don't know what a school is, if you don't know what a bus is. And this is what often we encounter in biology or biomedical research. We don't really understand the mechanism. So it's a challenge to actually look at the right moment, which could be, for example, the interaction of an immune cell with a target cell, or the event where a tumor cell invades the tissue, and understanding how it does that and how we can prevent it. So with that in mind, the first part of my talk is going to be about using the two-dimensional pair correlation functional analysis to study the spatial dynamic organization of immune cell receptors. And the immune cell we will be investigating is the natural killer cell, or NK cell. This is represents about roughly 10% of your white blood cells. Most of you are probably more familiar with the T cells and B cells. The B cells represent the adaptive immune response producing antibodies, whereas the natural killer cell is part of the innate immune response that can act immediately as a kind of first line of defense towards tumor or virus infected cells, for example. And they do this by exhibiting spontaneous cytotoxicity toward those abnormal cells. But during the response, they can also rapidly secrete cytokines and other chemokines to communicate with other immune cells and, for example, regulate the T cell response and the following adaptive immune responses. So if you want to say what's good about the NK cell, well, it can immediately get rid of some cancer cells or virus infected cells. However, thinking about medicine, advanced medicine, there's also an issue, which is if you're thinking about organ transplants, then the NK cell is a cell that attacks and rejects those kind of foreign entities. So these patients usually have to take very strong immunosuppressants, which then can lead to other problems. So the aim of the study is to understand the NK cell receptor organization and dynamics that may help improve cancer treatment and organ transplantation. And I also want to point out that this study was done in collaboration with the group of Sophia Johansson at the Tumor and Cell Biology Institute at the Karolinska Institute in Stockholm, Sweden. Okay, so a brief introduction about the function of the NK cell. So on the top left, we have a schematic representation of our NK cell, and on their surface, they express certain receptors, and one category of those receptors are the inhibitory receptors. And then all the other cells in your body, they, on their surface of the cell membrane, they express the so-called major histocomplex 1 molecules. And those are unique to the specific organism. And those are the molecules that the NK cell looks for. So if it finds those on the surface, it will most likely decide that this is a normal healthy cell and then will just continue its way. However, if these inhibitory receptors are missing, or if they have the wrong kind of form of inhibitory receptors, then the NK cell will notice this and can induce lysis by secretion of those cytokines and thereby destroy the abnormal cell that is missing those major histocomplex 1 molecules. So how does the NK cell know which kind of receptors to look for? Like us, the NK cell has to be educated to be able to distinguish those cells. So what usually happens is that the NK cell starts as a precursor cell, followed by stochastic expression of multiple different kinds of inhibitory receptors. And at this stage, the NK cell explores its environment and then tries to dock with other cells and thereby learn what is considered a normal cell in this environment, or a healthy cell in this environment. And when this process is complete, we say that the NK cell is educated and then can perform the function that it's supposed to do. However, the process can also fail. Maybe the NK cell is never able to express the correct receptors to identify those normal cells. And in this case, the cell will become hyper-responsive or stay non-educated. And it's very important that this cell will stay non-active, because going back to the previous slide, if it's expressing the wrong receptors, it would consider every cell in its environment an abnormal cell, and then basically randomly destroy tissue. So it's very important. This process of education is very important for the function of the natural killer cell. Okay, so this was the most simple model, only considering the inhibitory receptors. In reality, it's more complicated. In addition to inhibitory receptors, we also have activating ligands. So if a cell is in distress, it can display those activating receptors on its surface, or ligands, I mean, on its surface, and thereby also trigger activation of the natural killer cell. And oftentimes it's a balance between the ratio of inhibitory and activating receptor that determines if a natural killer cell will respond or not. So how can we distinguish those educated from uneducated cells? Well, you might imagine that there must be some kind of specific difference on the molecular level, which is true in a way, but these differences can be really subtle and can be very hard to actually label or target or examine in a cellular environment. Because in general, both, doesn't matter if it's educated or uneducated, they both express activating and inhibitory receptors, just not the very correct ones in terms of the uneducated cells. So what we did in the study, we started with a mouse model, and one cohort of mice is sort of wild type. So they have a regular functioning immune system, and they have those educated natural killer cells. The other group is a modified genetically engineered type, where the major histocomplex molecules have been knocked out. And because there are no MHC molecules, the NK cells will never get educated. These cells are highly, these mice are highly immunocompromised, so you have to keep them in a very clean environment, as they can get infected with diseases very easily. So to investigate the NK cells from these two types of mice, we extracted the primary cells from the spleen, followed by fluorescence labeling using antibodies, and then imaging. And we were looking at both inhibitory receptors. In this case, it was the LY49A, and also activating receptors, in this case, the NKP46. For imaging of the primary cells, we chose total internal reflection microscopy, which is a method that exploits the change in refractive index between a cover glass and the aqueous environment of the cell to limit the excitation depth to a few hundred nanometers. So we can get very good signal to noise at the cell interface with the cover slip at the membrane where the receptors are. And we can acquire images very fast using a camera. So here are some exemplary images of those cell contacts with the surface. The block that you can see on the left are the regular cells from the regular mice, so they may be educated. And on the block on the right are the ones where the MHC is knocked out, so they're all uneducated. The top row with the green color code represent the activating receptor that had been labeled with green fluorophore. The middle row, the inhibitory receptors labeled with a red fluorophore. And the bottom row is just the merge of the two images, and you can see that the receptors activating and inhibitory receptors are in different places. But then the question is, well, from those images, can we tell any difference between the educated and uneducated cell? And looking at many of those pictures, we were not able to see any particular patterns that were different. So no differences in the average intensity patterns observed. But then the question is, these are living cells and these are undergoing dynamic changes. So what about the dynamic spatial organization of those receptors? So this is what we investigated in the study. We were using, let me go back to the slide once more. So you can see that there are these clusters, and we were using single particle tracking and also fluorescence fluctuation methods to determine diffusion coefficients and binding times. But here in this talk, I want to focus on the two-dimensional pair correlation function approach, which has the unique ability to map barriers in the cell, as you shall see. Okay, so what we did is we record images of this cell contact or immune synapse very fast. So many planes as a function of time, typically on the order of 5,000–6,000 images at several tens of frames per second. And then in those stacks, what we do for analysis, we pick a pixel. So let's say the pixel marked in blue here, indexed Ri. And then we correlate it to a neighboring pixel at a certain distance, delta R, using the correlation function where the formula is shown here below. And so what do we get there as a function of the delay time? Well, first the autocorrelation amplitude increases, and then over longer times it will go down to 0. So what does that mean? Well, what we're looking at are fluctuations caused by molecules that are moving around while we are taking this image time stack. So what will happen, we have a molecule at position Ri, which then as a function of time can diffuse or migrate to the position Rj, which means it increases the likelihood of finding it over there. So the correlation amplitude first increases, but then the molecule will move further away from both of these points and eventually be so far away that the overall probability to find it in the same spot goes down to 0. Okay, however, this is only one pixel pair. So what we can do in a second step is we can repeat this correlation process for all pixels at a certain distance surrounding the center pixel of interest. So we get many of those decay curves, maybe 16 or 32, depending on the parameters of the algorithm, that we can then represent as this polar two-dimensional plot, where we have correlation amplitude as a function of lag time, and then for all the different angles around the center of interest. And we call these kind of mini images, we call those a sprite. So this is for only one point of the image. Now, what we do is we repeat this process for all the points in the image. So for each pixel, we get one of those little 2D images, one of those sprites. So as you can imagine, there are quite a lot. If we record an image of, let's say, 256 by 256 pixel, then we have 65,000 something of those little sprite correlation functions. Okay, so we have used an algorithm to generate a lot of data, but what do we do with this kind of data? So now we have to think about what happens to the particles that cause the shape of these correlation functions. So the first easiest case is we have molecules that are freely diffusing and isotropically, so have no preferred direction. All the directions are equivalent. So this means that in any direction, so at any angle, we will get the same kind of correlation function because the probability to find a molecule will be the same. So we get a symmetrically shaped form of the sprite. So no long axis or short axis. So the size is the same in all directions. However, if we happen to look at a pixel where we have molecules that are near a barrier, near an obstacle, then they can move in certain directions, but are unable to move in one direction, which would be here over the barrier. Or at least it would take them a very long time or longer time to cross over the barrier to get this other area. And this will cause a deformation of our sprite. So now we have an axis that is longer versus another axis that is shorter. So we can exploit this geometry to map out how the molecules move in our sample. So to illustrate this, here the green spots in this box just represent those molecules that can move and that we have labeled using a fluorescent marker. And then we have a barrier, in this case, this red S-shaped line. However, in the real sample, we cannot see the line. So it's unlabeled and it's very thin. We only see the molecules. But near the barrier, you will see if we do the calculation and the sprites, they will be deformed because the molecules are unable to cross this barrier. And then to quantify this, we can define certain, for example, a ratio of the long axis versus short axis, where if this becomes large, that means we have a large difference between the long and the short axis, what we call a high anisotropy, indicating that there is some sort of barrier near this pixel. So we can create a map of pixel showing the path of the molecules. So we simulated this. So the first simulation was using a grid. So the red lines again are barriers that the molecules cannot cross. And then we seeded molecules in those compartments at equal concentrations that we then let diffuse at 1 square micron per second, simulating many frames. And then the next picture, trimmed intensity, is just basically the average intensity of all those molecules. And as you can see, you cannot see any structures because, again, the barriers are very thin and unlabeled. And because the concentration of the molecules is the same more or less everywhere, we cannot see any differences. However, if we apply the algorithm, you can see we come up with this anisotropy map, and we can immediately see where those barriers are. And what you also see is that we see those double structures along the double line along each of the barrier. The barrier itself is only a single line, a single barrier. However, to explain this, so the molecules are moving and bumping in one compartment to one side, let's say to the right, and then on the other side, the molecules are bumping into the left. So this is why we get this kind of double feature, and also for up and down. There are other parameters that we can define. Let me go back one slide. So instead of the ratio of the two axes, we can also quantify the angle of, let's say, the short or the long axis. And this is done in this so-called connectivity map, where we plot lines, and those lines are the length that we plot. So the vector length is corresponds to the anisotropy we measure, and the orientation, so the angle of the line, corresponds to the angle of this sprite. And we color-coded the line with this color scheme from 0 to 180 degrees, where 0 and 180 has this reddish pinkish color scheme, and the 90-degree has the greenish color. And you can see with the, of course, it's a simulation, so we are expecting to get a perfect result. We simulated another example that can happen in biology, which are sort of tethering points where molecules can get temporarily trapped. Again, in the intensity, just the intensity information, this is very hard to see because all the signal of the molecules overlap. We cannot separate them and we cannot see where they're going. If we apply the algorithm, we clearly see those tethering points in the anisotropy map. And again, the connectivity map is kind of able to tell us the flow of the molecules around the barriers. Okay, so this is applied to our natural killer cells. We are having the, on the left-hand side, we are having the educated cells. On the right-hand side, the uneducated. Again, top is activating receptors, and the two bottom rows are two different kinds of inhibitory receptors. The top is activating receptors, sorry. And these are just examples, and you can see a clear difference. For the educated cell, we have kind of a medium amount of barriers. For the activating receptors, we have a lot of barriers for the uneducated cell for the activating receptor. And the inverse is happening for the inhibitory receptors. And we can quantify this and see that there are significant differences between the educated and uneducated cell. One little remark, you see that for the educated, the size of the distribution is always larger than the uneducated. And the reason is that all knockout model NK cells are uneducated, but the wild type mouse NK cells can be both uneducated or educated. So there we have a heterogeneity of the population, and this is why the distribution is broader. So this prompted us to come up with a model. So our hypothesis is of what's happening is that the activating receptor is less restricted in educated natural killer cells to facilitate binding to target cell antigens. So if we want to make this identification process quick and efficient, we need to make sure that the receptors are flexible to move, to dock to the targets. Whereas in the uneducated cell, for the activating receptor, we don't want this. So it's better to have a rigid, inflexible structures. And the inverse is true for the inhibitory receptor. Okay. So this leads me to a general approach that is taken in biomedical research, which is the one we were also doing. So you start off with an animal model. However, it's very difficult to image on the molecular level inside an animal. So what we usually do, Well, of course, you can do tissue slices, but then everything is fixed, so you cannot look at dynamics anymore. In our case, we extracted the primary NK cells and then transferred them to a culture dish where we could label and image them. However, this approach is limited because we cannot guarantee that the NK cell will behave in the same way as in the animal. It's just an assumption that we have to take. So what people are trying to do, and us as well, is to try to move to a 3D cell culture model, or more advanced models are also often termed human on a chip models, where you have at least some sort of extracellular structures where you can have a model for a tissue in a three-dimensional environment that is more physiologically relevant. But then, contrary to the animal, this environment can be very controlled. We can do live imaging, and we can use those high-resolution modalities to quantify molecule dynamics. Okay, to move to this system, we also need a new type of microscope. And so the next part of my talk is going to be about the light sheet microscope, where we can do the fluorescence fluctuation spectroscopy method in 3D environments and also apply other multimodal imaging methods. Yeah, so with using the SPIM, you can do the classic FFS methods, IMSD, for example, to get diffusion coefficient binding times and average transport velocities. And then we can also do the barrier mapping using our 2D PCF approach. And this project was just got funded last month, so I'm very excited to get going on this. Okay, so part 2 is about the light sheet microscope. And here I want to focus on another imaging modality, which is hyperspectral imaging using the light sheet microscope. First, for you who are not familiar with the light sheet concept, generally in microscopy, there are two kinds of approaches. Of course, there are also hybrid approaches, but generally speaking, there's epi-illumination, where we have a large area detector like a camera, or it could also be your eye, for example. We have many image elements, pixels that are all detected in parallel. So the advantage is it's very fast. The problem, however, is that if we are illuminating the whole sample at the same time, we will get lots of background and no optical sectioning, and also subject the sample to a high dose of light. At the other end of the spectrum, we have single point methods where we focus the light into a point, and then to get an image, we either scan the sample or the beam and then reconstruct the image from that on the computer. Hereby, we can get very good optical sectioning by either introducing an aperture like a pinhole, or by exploiting nonlinear excitation such as two-photon processes. However, the disadvantage is that this is generally a slow imaging method. So the light sheet microscope, or also called selective or single plane illumination microscopy, uses two objective lenses, generally perpendicular to each other, and one objective is used to generate a sheet of light to illuminate the focal plane of the detection lens. Using this method, we can get excellent optical sectioning capability, a large field of view and depth, high light throughput, high temporal resolution, and low light exposure levels, because we only illuminate the plane that we're interested in. Here are some references, and if you look at the first one, is from 1903. So meaning this concept has first been introduced 117 years ago. And then not very much happened with this method until the 90s and early 2000s, where the next two publications came up. So why was that? Well, I talked about using cameras, using lasers, using computers to analyze the data. digital data processing. All of that was not available in 1903. So this is why just recently this method now has really accelerated and made an impact in the field. So here, I'm not asking you to read all the details. I'm just flashing the slide of all the methods that have been developed recently. And this is not a comprehensive list. So if you are using light sheet microscopy in your publication, is not on there. My apologies. And this slide is just intended to show that it really is going off in the field right now. Of course, we have our own concept for this approach, which we call the side spin, because as you can see in the picture here on the left, we have this custom sample imaging chamber where we deposit our sample, and then we detect from the bottom, and we generate the light sheet from the side here through this cover glass at a 90-degree angle. And our setup has multiple sources of excitation, and we can do spectral lifetime and FFS imaging, and I'll focus on the spectral imaging for the second part of my talk. And I'm here just to show you just how the setup looks like, the optical components. All the things to generate the light sheet are located on this platform to the right-hand side of the microscope. And we can do linear excitation, visible light, and nonlinear excitation using near-infrared light. Here are just some example images, just to showcase that you can image very fast in 3D using these methods. These are giant unilamellar vesicles where we induced phase separation by mixing different lipids. And then, of course, these are diffusing, and you can track those lipid rafts in 3D as a function of time at, in this case, 1 stack per second. Here's another example of cells where we labeled lysosomes. Again, we took Z-stacks. We took a total of 500 Z-stacks, 80 planes each at 4-second intervals. And you can very precisely track the movement of the lysosomes in XYZ. And on the right-hand side, you can see the histogram of the velocities. Here's a 3D movie of cells expressing EGFP paxillin. And paxillin is a focal adhesion protein that you see predominantly occurs in those adhesion complexes that the cell uses to grab onto other structures, in this case, the surface of the cover glass. And here we have an example of a zebrafish embryo where we looked at the microcirculation of red blood cells. And here the imaging speed was 5 stacks per second. So with the light sheet microscope, you can image 3D volumes at almost video rate, which is very nice. But I want to focus on hyperspectral imaging, which has important applications in biomedicine. Of course, giving this talk at the Beckman Laser Institute, you're the experts in hyperspectral detection and imaging. And you know that there are many important applications, including diagnosis and monitoring of tumors and tumor treatments, monitoring of hemodynamic activity, and image-guided surgery, and many more other applications. In terms of the light sheet microscope, previous work includes the application of spatial scanning to be able to detect spectral information. So we sacrifice one of the spatial dimensions of the camera to detect the different spectra using a prism or grating to split the light, and then we have to spatially scan to reconstruct the 2D image. Instead, we can continue taking the 2D image, but instead scan the spectrum using a tunable filter, for example. And then another group was using faster snapshot imaging, where we can get back to, we can again take advantage of the high imaging speeds of light sheet microscopy. However, here you have to separate your image into a set of sub-images, so you strongly decrease your field of view. Instead, our approach relies on the spectral phasor, which transforms spectral information in the frequency domain to do the analysis. So in the regular approach that you would apply this spectral phasor method is, okay, you have your sample, your microscope set up, you acquire a spectrum in each pixel that you can then transform to the phasor plot using the sine and cosine transformation here. And then the information represented in the phasor plot is the center of mass of your spectrum, given by the angle here on the plot in your detection range. So counterclockwise, in this case, from 400 to 700 nanometer, which was our detection range. And then the second coordinate is the distance from the center, which represents the width of the spectrum, so how broad it is, with a very narrow spectrum like a laser line being on the outside of the circle and infinitely broad white light source being on the center of the phasor plot. And then we can identify components and project this information on our 3D image to create this four-dimensional hyperstack of information. So how, well, this is just a method for analyzing the data, but how can this be improved? How can this improve our light sheet imaging capabilities? Well, we don't necessarily need to acquire the spectrum and then do the transformation. Instead, we can use an optical element to directly do the transformation of the photons detected. So we are using filters shaped like a sine and a cosine function. So the transmission of the filters follows the shape in our detection range. And what we get from that is we directly get this frequency domain spectral phasor information without actually having to acquire the spectrum. And so because we only need two filters to do so, yeah, the sine and cosine filters, here you see the spectrum again in combination with the bandpass to define our detection range. We can get this information without having to go through hardware and splitting of the light and scanning methods to reconstruct the spatial and spectral information. So here, just to showcase that it works, we compared with a conventional 32-channel spectral detector, where the spectra were transformed in silico to the phasor plot. Here, different dyes, as an example, where they locate on the phasor plot, and then we compared the phase, so the center of mass of the emissions, the 32-channel against the sine cosine filters, and we see that there's very good agreement, and the same is true for the modulation. So for the distance here to the center of the phasor plot, so it's in very good agreement. And also we validated the rule of linear combination, which is a very important property of the phasor plot. So you have, for example, two components, can be more than two, but the rule of linear combination says that any mixture of the two must locate on a connecting line between the pure species. And depending on where they occur on the line, we can actually calculate the ratio of the two, which is very handy for analysis in biological samples. Okay, to introduce back the spatial information, we imaged 5 species of 1 micron fluorescent particles. On the left-hand side, you can see the emission spectra. Then in the middle, we see the phasor plot, and you see where they localize differently. And then we just, off of the phasor plot, we color code according to the different sections in blue, green, yellow, orange, and magenta, to identifying the different components and then mapping back to our 3D stack to generate the 4D hyperspectral image. Okay, to further investigate how the method works in a complex biological sample, we turn to the zebrafish model, which is a very popular model to study developmental processes. And we were looking at the zebrafish retina. And here the idea is we have different genes expressed with target fluorophore, RFP, GFP, YFP, and CFP. And these are expressed in one or multiple cells at different levels, depending on what the cells develops into or differentiates into during the embryo development. And this difference in the ratios of the fluorophores kind of creates a unique spectral fingerprint for each cell type that is difficult to separate just using the conventional bandpass filter methods, but as you will see, very easy to differentiate using the hyperspectral light sheet phasor imaging. So here we have, on the left-hand side, we have an intensity image of the eye of a zebrafish, the top view of this 3D stack. In the center, we have the phasor plot. So now you can see we have this large distribution. We don't have separate points like for the beads, and the reason is because all the species are intermixed in the different cell types. However, we know the species that we have. We have CFP, GFP, a nuclear stain, cytox, orange, and we have the red fluorescent protein indicated here with the different colored dots. And then in the reconstructed 4D image render, we just have them color-coded according to the gradients along the connecting lines between GFP cytox and CFP RFP. Here's the region highlighted here by the white box zoomed in. And these are the different colors that we chose based on the phasor plot. And by taking the profile in the different regions that we chose, we can see that it nicely matches the model of the photoreceptors and the rest of the structure of the retina that we expect. Okay, just a little bit more about the phasor plot. So one advantage of the phasor method is the reciprocity, meaning that you can go off the phasor plot and then select regions there and highlight them in the image, but you can also choose regions in the image based on the intensity and then reconstruct the phasor plot from that. And this is what we have done here, highlighted the different regions representing the eye architecture. And I don't want to go into too much detail, but you can see that every of those regions has a unique spectral fingerprint on the phasor plot that we can distinguish and then utilize to identify the different purposes and biology of the cells that is going on. We can also do, using the speed advantage of the light sheet microscope, we can do organelle tracking in live 3D cultured cells. We labeled them with 4 different dyes, labeling the nucleus, Golgi, mitochondria, and lysosomes. You can see a single reconstructed 4D hyperspectral image. And then here we have the 5D hyperspectral stacks as a function of time. And again, like you saw in the example in the beginning, we can track individual lysosomes as a function of time and separate them out from other tissue components or cellular organelle components. Finally, one application in live mouse tissue, ex vivo intestinal crypts. And here we didn't use any labeling at all, but instead we relied on the intrinsic fluorescence of the metabolites NADH and FAD. And depending on the kind of metabolism that the system is undergoing, which could be oxidative phosphorylation, for example, or glycolysis, the ratio of NADH to FAD will change. So you can define this as the optical redox ratio, NADH over intensity of FAD. and thereby can get some information about the underlying metabolism of the sample. And we chose the intestinal crypt because in the intestinal crypt, as the food also, unfortunately, the cells are digested as well in the intestine and they have to be replaced. So there are crypts that have a stem cell population where they can regenerate the differentiated cells in the lumen. And if you look at the metabolism, it turns out that the stem cell population is preferentially undergoing glycolysis and the differentiated cell population is preferentially undergoing OXPHOS, which you cannot see in just the intensity information. So here on the right, we have the 3D rendered structure of the tissue, just fluorescence intensity, and you cannot see any difference. However, if we apply our Phasor hyperspectral imaging method, we can see a gradient along the axial direction representing the stem to the differentiated region. And on the right-hand side, we have quantified the redox ratio from the Phasor, and you can see in XY the changes. There are not many changes at all. It starts more or less at the same value as it ends, but in the axial direction in Z, on the one end, we have this high redox ratio indicative of the stem cell population, and at the other end, in the lumen, we have the differentiated cell population. Okay, so with this, I want to summarize and connect back to the two problems in biomedical imaging that I mentioned earlier. The general conflicts whenever you do any imaging is ideally you want to get as large of an imaging volume as possible, but you also want to get as high a resolution as possible, high spatial resolution, high time resolution. But then again, the events can be sparse in both time and space, so there's always this conflict between getting high resolution and probing a large volume of interest. So this is why I want to use the Lightsheet platform as a very flexible multi-parameter imaging approach to try to help with this problem. And the idea is instead of the self-driving car, eventually maybe we will have something like the self-driving microscope, where we can have tissue samples in many wells that can be even perfused through a fluid inlet. And then the microscope can do a low resolution, large field of view screen of the individual tissues, and then identify certain events of interest, for example, based on spectral information, and then be able to automatically zoom into this region and then record it at this event with very high temporal, spatial temporal resolution, so that you, for example, can apply the fluorescence fluctuation methods that I talked about earlier. And then after the event is complete, it can resume sort of this monitoring scanning stage to identify the next event. So a lot of people are throwing in the keyword big data. And to that I say, maybe we should do smart data. If I'm very honest with myself, most of the data that I have acquired on the microscope is actually just noise, because it's exactly this problem that I wasn't able to identify the right cell or event of interest that we were looking for, and thereby using a combination of all these concepts, I hope that in the future we will be able to improve on that. With that, I just want to quickly introduce some other projects, just one slide each. I decided to go into this one or two projects in a little bit more depth instead of just trying to cram in as much projects as possible. But I just want to say that we can also horizontally integrate those techniques, for example, using the lifetime and or spectral sine cosine filter detection for in vivo imaging using a fiber optical device. And we can integrate these methods using our particle counter and sorter for diagnostic applications of detecting rare particles in large fluid volumes, such as blood, for example. Finally, I want to point out a very recent project that started in collaboration with the UCI Vaccine Research and Development Center and Zhao Lab for serological testing for COVID-19. Where they have a microarray with many different antigens where they can test your blood and see if you have antibodies against the virus. And we were working on the imaging end of that. You might have recently got an email from the UCI where they invite you to participate in a study. So we are starting a 7,000 people study here at UCI to investigate the exposure of the community to this virus. All right, with that, I would like to thank, of course, the Laboratory for Fluorescence Dynamics, where I did most of my work and where also the setup is located at the light sheet setup. Especially I would like to thank my postdoc mentor Enrico Graton, and then of course other group members. There are many more members I'm just mentioning who has helped me with the project I presented. Michelle, Lionel, Suman, Sasha, Rachel, and Hontao. Then I want to thank my collaborators at the Karolinska Institute, Sophia Johansson, Eilina, Sunita, Joachim, and other labs at UCI for supporting me logistically and with samples, including the Waterman Lab, Schilling Lab, and Zhao Lab. And finally, also the Institute Pasteur in Uruguay, Montevideo, for providing the sofafish zebrafish line for the hyperspectral imaging in the zebrafish. And last but not least, of course, funding. The LFD is supported by a P41 resource. We got some funding from CCBS through an opportunity award. Thanks also goes to the Swedish Research Foundation. Then there's the New Art 21 I mentioned earlier, and then support from UCI for the other two projects, the Cancer Center project and also the UCI Craft COVID Award. So with that, thank you very much for joining today's seminar. And yeah, please let me know if you have any questions. I'm happy to answer.
Thank you, Nick. That was a great presentation. Excellent work.